So in this section, I'm going to pretty much focus on the systems that we have available within our platform. We by no means have every single system that's available. That would become very, very expensive. But I think it's a good starting point, at least, to make you aware of some of the applications that you can do using fluorescence microscopy. Okay, and the way I'm going to run this is looking at imaging across, using microscopy across the imaging scales. So from tissue or model organisms on the right-hand side here, through tissue to cells to subcellular, down to molecular, and then finally into the single molecule range or the atomic range. But I wanted to highlight that fluorescence microscopy can't get you this entire range. So in red here, we've got the regions where standard fluorescence microscopy will get you. That takes you down to about 250 nanometers resolution. And this is what we call the diffraction limit. So that means that based on the wavelengths of light that we're using, we can't resolve structures that are smaller than 250 nanometers. If you want to do that, then we can use a group of techniques called super resolution microscopy, which again can use visible light and a few interesting tricks to get around this diffraction limit. That will take us down to about 20 nanometer resolution. And if you want to go beyond that, then you're going to have to look at a different approach, something like electron microscopy. So in this lecture, I'll cover the fluorescence microscopy and the super resolution microscopy. If you want to learn more about electron microscopy, that is not my area. Okay, so we'll start up on the right-hand side with larger pieces, and we'll move down through a number of the different applications that are possible. Okay, so the ones we're going to cover are light sheet microscopy, multiphoton microscopy, slide scanners, high content imaging, and again, wide fields microscopy. And that will take us through the kind of organs, tissues, and cells regions of this, of the scale of imaging. Okay, so light sheet microscopy is regularly used in developmental biology and neuroscience applications. It's a very macro level imaging, so mostly kind of cellular resolution. In this case, as the name suggests, your sample is illuminated by a light sheet. So in the figure down here, this is our specimen in green. In this case, we have two objectives. So we've got one objective which creates this very narrow sheet of light, which comes in perpendicular to our imaging objective. The nice thing with light sheet is that we're only illuminating what we're actually imaging. So the dotted red line here is our focus, and you can see we're only illuminating the sample where we're actually imaging. This means it's very, very good in terms of photo bleaching or damage to our samples based on the excitation line. We can then move the specimen through this, and that will allow us to build up a 3D volume of the sample. Very often these are used imaged using dipping lenses or through specially designed cuvettes, so that the lenses don't necessarily come into contact with the samples. The downside is that the sample needs to be clear. So these specimens can be anything up to like a centimeter in size. We regularly do mouse or rat brain imaging here, so we can image a whole model organism brain if you want. Commonly, they're used for, as I said, developmental biology, so looking at embryos of things like zebrafish or Drosophila, worms, things that are kind of essentially optically clear already. If you're putting a piece of brain or whatever in there, then we have to go through and chemically clear that tissue. So how do we do that? So this is a very old technique, was first developed in 1914, and there's essentially two main methods. We can use organic solvents, which essentially remove water from the sample and then match the refractive index with the organic solvent. The downside of this is it's not always applicable to fluorescent proteins. This can take a bit of time, can also kill off the fluorescence from the fluorescent protein. Or there are aqueous-based methods which can involve removal of lipids, and again, refractive index matching. These can be kind of sugar-based methods like CDB, or things like Clarity, where you embed your sample within a matrix. And this is an example up here of a brain pre-clearing and post-clearing with the Clarity approach. The downsides of tissue clearing is, first of all, it can be tricky to set up. The reagents can be very expensive. It can be a very, very long process in terms of actually clearing your samples before you get to the point of imaging. So it's something you want to make sure you really want to get into. It's not something you dip your toe into. This is one example from one of the researchers here at the University of Melbourne. This is a rat cochlear imaged in multiple channels. And here they're looking at the damage caused by cochlear implants into this spiral structure here within the cochlea.